So what do you think? Which of the following would have the highest conduction speed? Neurons that are myelinated with a small diameter, neurons that are myelinated with a large diameter, or unmyelinated small diameter axons, or unmyelinated large diameter axons? Well, the answer is B. Myelinated large diameter axons actually have the highest conduction speed. With a larger diameter, neurons actually have a lower resistance. This allows the action potential to move faster along the neuron. We see this in cephalopods like squids and also octopus. They have large diameter axons without any myelin on them that can actually conduct action potentials really fast, and they have really quick nervous system responses. One of the fastest, honestly, in invertebrates. If you're wondering how I got around today, well, since I didn't move, I took the stationary bike. Myelin can also speed the conduction of action potentials along the axon. Myelin is either caused by Schwann cells that wrap around the axon in the peripheral nervous system, or oligodendrocytes that wrap around the axons of neurons in the central nervous system. The myelin basically insulates part of the axon and causes the action potential to have to jump from one exposed region to the next exposed region. These exposed regions of membrane that can actually have gated ion channels are known as nodes of Ranvier. And so the action potential has to jump really from node of Ranvier to node of Ranvier. This is known as saltatory conduction, after really the Spanish word saltar for jumping of the nerve transmission along the axon. So why are action potentials usually conducted in only one direction along an axon? Read the question, guys, pause the video, and I'll tell you the answer. Okay, the answer is B. The brief refractory period prevents reopening of voltage-gated sodium ion channels and doesn't allow the nerve transmission to go in the opposite direction. We're going to take a look at this little short video here. They have some frog legs, and they're going to put salts on them. Salt, of course, is sodium chloride. Pause your video, guys, actually, and explain what's occurring in the frog legs. Okay, what's actually occurring is that the sodium is flushing into the neurons, causing an action potential to be passed along the neurons and causing then muscles to contract. In order for a neuron transmission to pass on from one signal to the next signal, it has to pass across a synapse. There are some synapses that are just gap junctions, and the neuron signal is passed directly, the action potential is passed directly from one cell to the next. But in most cases, the synapse is actually a space between one neuron and the next neuron. And from one space to the next, there has to be a chemical that's passed across. The chemical is usually referred to as a neurotransmitter. So usually the transmission across synapses going from one neuron to the next requires a few things. So first of all, the action potential comes along the axon and causes these voltage-gated ion channels, usually calcium ion channels, to open, allowing calcium to flow in down its concentration gradients and act as a second messenger, causing the exocytosis of neurotransmitters from the neuron. This is known as the presynaptic neuron because it's before this space. The space actually is referred to as a synaptic cleft. The neurotransmitters then move through diffusion to the postsynaptic membrane, the next neuron. And they usually bind to ligand-gated ion channels. These ligand-gated ion channels then open and allow ions to flow in. For instance, sodium might flow in and cause depolarization of the next neuron. So there are a few different types of neurotransmitters. Acetylcholine is one neurotransmitter that we're going to spend a lot of time on. There are amines, dopamine and serotonin, for instance, are these amines. They have amino groups at the end. You may have heard of these before. There are amino acids like GABA, glycine, glutamate, aspartate. These have both an amino group and a carboxylic acid. That's why they're amino acids. And then there are some neuropeptides that act like neurotransmitters. These are short polypeptides, really, that pass on the signal. Like I said, though, we're going to focus on acetylcholine. Acetylcholine is the neurotransmitter that's used to actually excite skeletal muscle. So in acetylcholine, actually, guys, it is released by neurons that innervate with muscles. Acetylcholine binds the ligand-gated ion channels and causes skeletal muscle to depolarize. We'll talk more about this later. Neurotransmitters also need to be removed in order to stop the stimulation of a postsynaptic cell. And they're removed in one of the following ways. Some of them actually may be retaken up by presynaptic neurons or surrounding glia and then degraded or reused. Some actually might be degraded in the synapse. For instance, acetylcholine is degraded by acetylcholinesterase, which is found on the postsynaptic neuron and degrades acetylcholine into acetic acid and choline. Some actually, neurotransmitters actually diffuse away and are degraded somewhere else, like the liver. Neurotransmitters can either cause an action potential or decrease the likelihood of there being an action potential. If a neurotransmitter actually depolarizes a membrane, it's known as an excitatory postsynaptic potential. If the neurotransmitter is actually inhibiting the next neuron, it's known as an inhibitory postsynaptic potential. Inhibitory postsynaptic potentials can hyperpolarize a membrane and prevent depolarization. Now, in order for an action potential actually to be produced, there needs to be enough of an excitatory postsynaptic potential to actually pass the threshold and cause an action potential to be passed down the axon. This can either be done by one neuron innervating with the postsynaptic membrane and exciting it really in close time. So you have two excitatory postsynaptic potentials in close time, then it can cause it to pass the threshold and reach an action potential. That's known as temporal summation. Or you could have two axon terminus releasing EPSPs, excitatory postsynaptic potentials, causing the action potential to reach a threshold, causing the depolarization to reach a threshold actually, and cause an action potential. Again, inhibitory postsynaptic potentials can actually counter any excitatory postsynaptic potential. Next up, we're going to talk about how neurons actually innervate with muscles. Hope everybody is staying happy and healthy. That is all.
Hi, I'm Zippy the Zebra. I'm your favorite neurotransmitter. My cousin is named acetylcholine. I'm a sodium ion. I'm a sodium ion too. I'm a sodium ion as well. I'm a sodium ion as well. Oh, I'm a neurotransmitter. Oh, sodium. Sodium. Go Team Trees.